And today we're going deep on something pretty fundamental to, well, to being human, really. It's all about how we experience the world, how we take it all in. We've got a really cool set of notes here all about sensation and perception.
Oh, yeah, this is a fascinating area.
And our mission, if you will, is to break down the basics of how we detect what's out there and then, and this is key, how we make sense of it all.
It's incredible, when you really think about it. I mean, there's just this constant barrage of information hitting us from all sides, and our brains are just, chugging away behind the scenes, just making it all make sense, creating this whole picture of the world from all that raw data. It's really, really impressive.
Yeah, for sure. So let's start with that first step, how we even detect what's out there in the first place. I mean, how do we even know there's something to make sense of? And that brings us to, I guess what you could call like a fundamental fork in the road, sensation versus perception.
Right. Two sides of the same coin, but definitely distinct.
Exactly. So think of sensation as that initial aha moment, right? Like your body's like, hey, something's out there.
Oh,
It's when those specialized neurons, those sensory receptors, actually pick up on some kind of stimulus. Like, boom, the alarm bells are going off.
and then transduction kicks in. That's where the alarm signal actually gets sent. It's the process, and it's, mind-blowingly elegant, where that stimulus energy, could it be light, sound, a chemical, anything really gets converted into an electrical signal, that action potential. And that's how your nervous system, your brain, can actually start to understand what's going on.
And of course, we've got this whole arsenal of sensory systems, each one fine-tuned for a specific job.
Yeah.
I mean, there's vision, obviously, but then there's hearing or audition, if we're getting fancy, and then smell. That's olfaction, taste, that's gustatiner.
Don't forget touch.
Oh, right, touch, which is really, it's like a whole category of physical feelings, pressure, temperature, all that. And we lump all those together under the very scientific sounding term somatosensation. And each of these systems, it's like perfectly designed to pick up on a certain type of information.
Absolutely. And the level of sensitivity in some of these systems is just, it's awe-inspiring. Take our vision, for example.
Which brings us to the absolute threshold. So this is the bare minimum amount of stimulus energy that's needed for you to actually detect something. We're talking 50% of the time here. And the example that always, I don't know, just blows my mind is being able to see a candle flame, a single candle flame from 30 miles away on a clear night, 30 miles.
That's pretty incredible. It really shows you how insanely sensitive those sensory cells can be.
I mean, it's really humbling, just thinking about the power of these systems we have.
Oh, absolutely. And speaking of what we can detect, there's this whole other area that's, well, a bit more mysterious, the realm of subliminal messages. These are stimuli that actually fall below that conscious awareness threshold.
Right. So they're kind of like these fleeting signals that might be triggering a response in your brain without you even realizing it.
It's like this hidden world of information processing that we're not even privy to. It makes you wonder what else is going on under the hood.
Right, right. And then we have the just noticeable difference, or J&D for short.
Yeah.
So this is the smallest change in a stimulus that you can actually detect.
And what's so interesting about the J&D is that it's not this fixed value, it's relative. It all depends on the strength of the initial stimulus. Think about your phone lighting up, for example.
Oh, yeah, good example. Like if you're in a movie theater, pitch black, even the faintest light from your phone screen is going to be super obvious. But if you're, I don't know, walking around a brightly lit shopping mall, that same amount of light change might be totally invisible to you. Same change, different perception.
Exactly. It's because our senses are always adjusting to the baseline level of stimulation. And all of this detection, all the sensation, it's really just the starting point. The main event, the real show, is perception. That's where our brains really get to work.
Yeah, totally. Because perception isn't just about passively taking in information. It's about actively interpreting it, organizing it, making it into something that we actually experience consciously. It's like taking all those raw ingredients, those sensations, and cooking up a delicious meal.
And there are so many factors that come into play, so many things that can influence how we interpret the world around us. One of the big ones is sensory adaptation. Have you ever noticed that? How you just kind of stop noticing things after a while?
Yeah. Oh, all the time. It's like,
Yeah.
when you first walk into a room and there's a clock ticking, you hear it right away. But then after a few minutes, it just fades into the background. It's still there. The sound waves are still hitting your ears, but you just stop consciously perceiving it.
Precisely. Our brains are wired to pay attention to changes, to novelty. So if something is constant, unchanging, it becomes less important.
Yeah.
And our brains just kind of filter it out so we can focus on what's new, what might be more relevant for survival.
And then there's motivation, which is something I think we can all relate to. Like when you're really, really expecting a phone call and you think you hear your phone ringing, but it's not.
Oh, yeah, I've totally done that. Or when you're like walking down a dark street and you think you see someone lurking in the shadows, but it's just a bush. Our desires and fears can really shape our perceptions.
It's like our brains are almost creating what we expect to perceive. And that's where signal detection theory comes in, right?
Exactly. Signal detection theory is all about how our mental state, how alert we are, what we're expecting, all of that, how it affects our ability to actually detect a signal. It's not just about the strength of the signal itself, it's about what's going on in our minds.
And it goes even deeper than that. Our beliefs, our values, even our prejudices and expectations, they can all have a huge impact on how we perceive things. There's this really cool study where people who had positive attitudes towards low-fat foods actually rated those foods as tasting better. Like their pre-existing beliefs actually changed how they experienced the taste.
It's a great example of top-down processing. It's like our brains are using what we already know, what we expect, to fill in the gaps and create a more complete picture of the world.
And this whole idea of experience shaping perception, it goes beyond just our individual beliefs and gets into like our broader life experiences, our cultural
Oh,
background.
absolutely. You see this really clearly with the Muller-Lyer illusion.
Oh, right, that one with the lines that look like they're different lengths, but they're actually the same.
Exactly. And the fascinating thing is that people from Western cultures who are surrounded by straight lines and right angles all the time, they tend to be more susceptible to this illusion than people from cultures where the environment is more rounded, with more curves.
So it's like our everyday visual experiences actually shape how our brains process visual information on a fundamental level.
It's incredible. It's like our brains are constantly learning and adapting to the world around us, even at the level of basic perception. And it's not just a visual thing. This happens with all our senses.
So we're taking in all this information through waves, right? Light waves, sound waves. Before we get into the specifics of how each sensory system works, let's break down the fundamental properties of these waves.
Sounds good. Let's start with amplitude and wavelength. Amplitude is basically the height of the wave, from peak to trough, and it tells us how intense the stimulus is. The bigger the wave, the stronger the signal.
So a loud sound would have a high amplitude wave, and a dim light would have a low amplitude wave.
Exactly. Then you have wavelength, which is the distance between two peaks of a wave. And closely related to wavelength is frequency, which is basically how many waves pass a certain point in a given amount of time. The shorter the wavelength, the higher the frequency, and vice versa.
So with light waves, those exist on this huge spectrum, the electromagnetic spectrum. And the light that we can see, the visible light, is just this tiny little sliver of that whole spectrum.
Right. And within that visible spectrum, different wavelengths correspond to different colors. That's why we see a rainbow. When white light is split through a prism, it's separating out those different wavelengths.
And with sound waves, the frequency determines the pitch. So a high-frequency sound wave would be high-pitched, like a whistle, and a low-frequency sound wave would be low-pitched, like a bass drum.
And the range of frequencies that humans can typically hear is from about 20 to 20,000 hertz.
Hertz being a measure of cycles per second. And then going back to amplitude with sound waves, amplitude determines the loudness. So a high-amplitude sound wave would be loud, and a low-amplitude sound wave would be quiet. We measure loudness in decibels, or dB.
Right. And it's important to note that exposure to sounds over 85 decibels for prolonged periods can actually damage your hearing. So those rock concerts you mentioned earlier, yeah, those can be a bit risky for your ears.
Definitely something to be mindful of. Okay, so we've talked about the general properties of waves. Now let's dive into how our visual system actually processes light waves.
So the eye is an incredibly complex organ. But in simple terms, light enters the eye through the pupil, and it's focused by the lens onto the retina at the back of the eye.
And the retina is where the magic happens, right? Because that's where the photoreceptor cells are.
Exactly. There are two main types of photoreceptors, rods and cones. Rods are super sensitive to light and allow us to see in low light conditions, but they don't really perceive color. Cones, on the other hand, are responsible for color vision, and they work best in bright light.
So when light hits these photoreceptors, it triggers a whole cascade of chemical reactions that ultimately generate electrical signals. And those signals travel down the optic nerve to the brain.
And there's this really cool crossroads called the optic chiasm, where some of the nerve fibers from each eye actually cross over.
Oh, right. So information from the right visual field goes to the left side of the brain and vice versa.
Exactly. And ultimately, all that information ends up in the occipital lobe at the back of the brain, which is the primary visual cortex.
And from there, the visual information gets sent off to different parts of the brain for further processing. One of the key pathways is the what pathway, which helps us identify objects.
Right, so if you see a dog, the what pathway is what tells you that it's a dog and not a cat.
And then there's color vision, which, as you mentioned, is primarily handled by the cone cells in our retinas. There are three types of cones, each sensitive to a different range of wavelengths: red, green, and blue.
And it's the combination of signals from these different cones that allows us to see the full spectrum of colors. But sometimes people have issues with one or more types of cones, and that can lead to color blindness, where they have difficulty distinguishing certain colors.
And then we have depth perception, which is our ability to see the world in three dimensions. How does our brain do that with just two eyes?
Well, it uses a bunch of different cues. Some of those cues are monocular, meaning we can perceive them with just one eye, like linear perspective, where parallel lines seem to converge in the distance.
Oh, right, like when you're looking down a long road.
Exactly. That's a cue that tells our brain that things are getting farther away. Then there are binocular cues, which require both eyes, like retinal disparity, where each eye actually sees a slightly different image because they're in slightly different
So
positions.
it's like the brain is combining those two slightly different images to create a 3D picture.
Exactly. And it's all happening automatically without us even having to think about it.
Our brains are pretty amazing. Okay, so that's vision. Let's move on to hearing, or audition.
So the ear is another incredibly intricate organ. It's divided into three main parts: the outer ear, the middle ear, and the inner ear.
The outer ear is the part we can see, with the pinna and the ear canal.
Right. And at the end of the ear canal is the eardrum, also known as the tympanic membrane. That's the boundary between the outer ear and the middle ear.
And the middle ear is where those tiny bones are, the ossicles.
Exactly. There are three ossicles: the malleus, the incus, and the stapes. And their job is to amplify the vibrations from the eardrum and transmit them to the inner ear.
And the inner ear is where the cochlea is, that spiral-shaped structure.
Right. The cochlea is filled with fluid, and inside the cochlea is the basilar membrane, which is the key to hearing.
So when a sound wave enters the ear, it travels down the ear canal and causes the eardrum to vibrate. Those vibrations are then amplified by the ossicles and transmitted to the oval window, which is a membrane-covered opening to the cochlea.
The vibrations of the oval window create pressure waves in the fluid inside the cochlea, which then cause the basilar membrane to vibrate. And along the basilar membrane are these tiny hair cells, which are the sensory receptors for hearing.
So when the basilar membrane vibrates, it bends those hair cells, and that bending triggers those hair cells to send electrical signals down the auditory nerve to the brain.
And those signals travel to the auditory cortex in the temporal lobe, where the sound is perceived. Now, how do we perceive different pitches? There are a couple of main theories.
One is the temporal theory, which says that the frequency of a sound is coded by the rate at which the neurons fire action potentials.
Right, so a high-frequency sound would cause the neurons to fire more rapidly than a low-frequency sound. But there's a limit to how fast neurons can fire, so this theory doesn't quite explain how we perceive very high pitches.
And that's where place theory comes in. This theory says that different locations along the basilar membrane are more sensitive to different frequencies.
So the base of the basilar membrane is most sensitive to high frequencies, and the tip is most sensitive to low frequencies.
And it turns out that both theories are actually at play. The rate of firing is more important for lower frequencies, and the place of stimulation on the basilar membrane is more important for higher frequencies.
And then we have sound localization, which is our ability to figure out where a sound is coming from. We do that by comparing the sounds that reach each ear.
Like if a sound is coming from your right side, it'll reach your right ear slightly before your left ear, and it'll also be slightly louder in your right ear.
And our brain uses those tiny differences in timing and loudness to pinpoint the location of the sound.
Now, unfortunately, many people experience hearing loss. There are two main types: conductive hearing loss and sensorineural hearing loss.
Conductive hearing loss is usually caused by a blockage or damage in the outer or middle ear, which prevents sound waves from reaching the inner ear properly. This can often be treated with medication or surgery.
And sensorineural hearing loss is caused by damage to the hair cells in the cochlea or to the auditory nerve itself. This type of hearing loss is often permanent, and it can be caused by exposure to loud noises, aging, or certain medical conditions.
It's a good reminder to protect your hearing by wearing earplugs in loud environments and keeping the volume down when you're listening to music. Now, let's move on to taste, or gustation.
Taste is fascinating. We used to think there were only four basic tastes: sweet, sour, salty, and bitter.
But now we know there's at least one more, and possibly two. We've added umami, which is that savory, brothy taste that's often associated with MSG.
And some researchers believe there might be a sixth basic taste for fat.
So our taste receptors are located in taste buds, which are these little clusters of cells that are mostly found on the tongue, but also in other parts of the mouth and throat.
And each taste bud contains a bunch of taste receptor cells,
hmm
which have these tiny hair-like projections called microvilli.
And when those microvilli come into contact with food molecules, it triggers a chemical reaction that sends signals to the brain via the gustatory nerve.
And the brain interprets those signals as different tastes. Now, smell, or olfaction, is very closely related to taste.
Right. In fact, a lot of what we perceive as flavor actually comes from our sense of smell.
So when we eat something, the odor molecules travel up from our mouth into our nasal cavity, where they're detected by olfactory receptor cells.
And those receptor cells send signals to the olfactory bulb, which is part of the brain, and that's where we perceive the smell.
Now let's talk about touch, or somatosensation. Our skin is our largest sensory organ, and it's packed with all sorts of receptors that allow us to feel pleasure, temperature, pain, and vibration.
We have Meissner's corpuscles, which are sensitive to light touch, and Pacinian corpuscles, which are sensitive to deep pressure.
And Merkel's discs, which are sensitive to sustained pressure, and Ruffini corpuscles, which are sensitive to stretching of the skin.
And then we have free nerve endings, which are responsible for sensing pain and temperature.
And all of these receptors send signals to the brain via the spinal cord.
And the brain interprets those signals as different types of touch sensations. Now, beyond the five basic senses, we also have some other important senses that help us navigate the world.
Like the vestibular sense, which is our sense of balance and spatial orientation.
The vestibular system is located in the inner ear, right next to the cochlea. It consists of these fluid-filled sacs and canals that contain hair cells.
And when we move our head, the fluid in those sacs and canals also moves, which bends the hair cells and sends signals to the brain about our head's position and movement.
And that information helps us maintain our balance and coordinate our movements.
We also have proprioception, which is our sense of where our body parts are in space.
Proprioceptors are located in our muscles, tendons, and joints. They provide information about the position and movement of our limbs and body.
And kinesthesia, which is our sense of movement.
Kinesthetic receptors are located in our muscles and joints, and they detect changes in muscle length and tension.
So all of these senses work together to give us a complete picture of our body's position and movement in space. And all this information is constantly being processed by our brains, and it's all happening automatically without us even having to think about it.
It's really remarkable. Now, one of the key ways our brains make sense of all this sensory information is through the Gestalt principles of perception.
The Gestalt principles are basically a set of rules that describe how our brains tend to organize visual information into meaningful patterns and shapes.
One of the main principles is figure ground, which is our ability to distinguish an object from its background.
Like that classic vase faces illusion, where you can see either a vase or two faces, but not both at the same time.
Right. Then there's proximity, where we tend to group together objects that are close to each other.
And similarity, where we group together objects that look alike.
And continuity, where we tend to see continuous lines and patterns even when they're interrupted.
And closure, where we tend to complete incomplete shapes in our minds.
All of these principles work together to help us make sense of the visual world around us. And there's just one example of how our brains actively construct our perception of reality.
So we've covered a lot of ground today. From the basic properties of sensory stimuli to the complex ways our brains interpret and organize that information. We've explored the amazing capabilities of our sensory systems and the factors that can influence our perception. And we've seen how all of these processes work together to create our unique experience of the world.
It's a truly fascinating area of study, and it really highlights how much is going on beneath the surface of our awareness.
Absolutely. So to our listeners, I encourage you to think about a time when your own expectations or motivations might have shaped your perception of an event. How did your pre-existing beliefs or your emotional state influence what you saw or heard? And how might understanding these perceptual processes help you to be more aware of your own biases and to better understand the perspectives of others?